Thank you for the very kind introduction. What we can do nowadays in medicine is pretty amazing. But when we enter a clinic, we should remind ourselves that what we see is only the tip of the iceberg. Doctors can only do as much as the resources allow them. Resources such as vaccines, diagnostics, and medical devices. And of course, drugs. Now, developing a drug takes often years, if not decades, to achieve in numerous research teams. And that itself is based on a tremendous amount of basic research that has been performed around the world. So we have this nice expression of standing on the shoulders of giants. If we want medicine to advance further, we have to have persons that are ready to be shoulders for others. And that's who we are. Hello, my name is Raphael Franzini. I'm an assistant professor in the Department of Medicinal Chemistry. What my group does is pursue a bottom-up approach towards drug development. We're starting off with basic science, trying to develop new chemistry, advancing new technologies to discover and deliver drugs, applying this to find new candidate molecules with the hope that ultimately they will be translated into the clinic. Broadly defined, my lab can be divided into two parts. One of them is to develop new chemistry that can be applied in vivo for delivering drugs. And the second one, to advance a technology called DNA-encoded libraries and use it for finding new drug candidates. In the interest of time, I will focus on the second part. But before I go ahead and jump into research, I want to tell you a little bit about myself. So I grew up in a blue-collar family in rural Switzerland. The picture that you see down there is an actual picture of where I'm from. And what brought me to this career is really my enthusiasm for science. I was the nerdy kid looking at the sky, the starry sky and wanting to know everything. So it very early has been clear that I wanted to be a scientist. Well, there's many different scientific disciplines. I've decided to be a chemist. And what excites me about chemistry, it's a creative science. Most scientists dissect nature in order to understand it. But in chemistry, very often you take two concepts, bring them together, and generate something new. And of course, it's an applied science. We're trying to advance mankind by doing science. So a lot of my training and education has been a back and forth between Switzerland and the United States, starting off studying at Lausanne chemistry, then coming to the US as an exchange student at Carnegie Mellon, where I was first doing active research, returning back to complete my master's degrees, to then again come to the US as a PhD student in the lab of Eric Kuhl at Stanford University, returning back for a postdoc at ETH with Dario Neri. And really what got me to go back was to wanting to learn this DNA-encoded library technology that Dario Neri has been one of the pioneers of. I recognize that this is one of the technologies that really has the potential to transform drug discovery. And that's exactly what is happening right now. So, having become an expert on DNA-encoded libraries, I was recruited to come here to the University of Utah to be part of our drug discovery community. So, there's many reasons that attracted me to come to the University of Utah, but one that really stood out is being in the College of Pharmacy, because the College of Pharmacy greatly epitomizes what I see as the type of science I want to do. It has the same trajectory of looking at basic sciences and gradually translating it towards the clinic. It's also at the interface of chemistry and biology, which is exactly where I would like my research to be. And of course, it's the core of any drug discovery activity here at the university. So one of the things that we're addressing is the bottleneck of drug development, which is to find new molecules. As I'm sure you all know, drug development is a lengthy process, taking multiple steps, starting from studying a disease, identifying a target, finding a molecule, optimizing it, evaluating it, moving it into clinical trials, and hopefully getting it approved. From a chemist's perspective, one of the key challenges is to find a molecule that binds to your target of interest and has the effect that you would like it to have. One of the standard approaches currently being used for doing this is called high throughput screen. I show you here a picture of the infrastructure that is used for an industrial high-throughput screen. And as you can see, this is a tremendous amount of infrastructure that is required. Briefly, high-throughput screens are expensive, they're laborious, and they really restrict our ability to screen targets broadly. Our approach, on the other hand, miniaturizes drug discovery. In our approach, we have libraries of small molecules that have an attached DNA code. And the DNA code tells us what the molecule is. So in this way, we can take a few microliters of our DNA-encoded libraries, add it to a well where we have a target immobilized, and then if there's molecules in the library that bind to our targets, they will be retained on the surface while we're washing away the non-binders. Now we can very easily identify what those molecules are by using the DNA sequence as a barcode that tells us what the molecules is, which we then synthesize and validate. This whole process that I've just showed you here takes about 2 hours and is really inexpensive to perform, opening up a lot of opportunities for studies that otherwise you wouldn't be able to do. So I've spent a lot of time trying to understand this technology and trying to make it better, but where we are right now is to take a step further and to use it for actual drug discovery purposes. I've become interested in the concept of aging. Age is one of the main, or is the main, risk factor for many chronic diseases such as cancer, cardiovascular disease, and neurodegeneration. Now there's many factors that contribute to aging, but one seems to be this molecule that I show in the middle, which is NAD. NAD regulates a plethora of processes, and as we get older, the level of NAD starts to drop, and these processes become dysregulated, leading to DNA not being properly repaired, metabolism, mitochondria not functioning properly, metabolism altered, and so on and so forth. So the idea is that if we understand those processes, we can identify targets and maybe modulate them with small molecules to address the underlying causes of diseases rather than treat the symptoms as they become apparent. To do this, we started off by making a DNA-encoded library specifically designed for NAD-binding proteins. And as a first step, we had to demonstrate that this library is working and that it is successful in identifying active compounds for protein targets that are in this area. And on this slide here, I show you a representative set of active molecules that we have discovered for such targets. But as I told you, we want to take this a step further and start to develop actual drug candidates. As of today, we're working on 3 targets. All of them are NAD binding. One of them is SARM1. SARM1 is regulating Valerian degeneration. And the idea is if you have an inhibitor to SARM1, you might be able to address neurodegenerative diseases such as ALS, multiple sclerosis, and maybe Alzheimer. And we have found active compounds for this target. Another target is SIRTWIN5 that is regulating metabolism, among other things. We have found molecules that activate SIRTWIN5, and we hope that we can translate those into new medications for kidney disease and cancer. And we're also working on activators of SIRTWIN6 that we would like to use to treat cardiovascular disease and again cancer. These projects are all in very early stages, but we have achieved a major breakthrough of identifying compounds that can be used for further development. This has been made possible by a very generous donation by the Alstom Foundation, which is a very major sponsor in the College of Pharmacy, and our SIRTWIN6 research gets funding from the National Institute of Aging. So with this, I want to finish up, and I started off by telling you about standing on the shoulder of giants. Of course, everything that I'm telling you here is me standing on the shoulder of giants. And with this, I would like to thank the persons that have done all this work and the funding sources that have provided us with support and our collaborator that have helped us along this journey. And I thank you very much for listening to my talk.